So I will do a simple introduction here. So generally that is Dr. Jessica Turner's lab, and we join them for the discussion of the paper. So from now on, we just kind of use your effort and extend more into this BSI discussion group. Hope more people can join the discussion. Generally, we are looking at is we have selected a top paper, which hopefully is interesting to all of you. Then we're going to discuss the paper and more of the open discussion, like we can share what we think we can learn from this paper, but also open to other questions like what we can learn and also how can we apply to our own research. So it's more of engaging instead of like one people to talk. It's more of engaging group discussions. And then we have Dr. Jessica Turner and also Nora. She is an excellent neuroscientist. We learned from her a lot. She's a big help to teach us what is the basic information of neuroscience. And myself is engineer background, so more of a method person. So I probably dig into all the mathematical part instead of neuroscience and psychology part. So, okay, without further introduction, and we can start the first paper.
Can I ask one more organizational question? We're going to do this roughly monthly, right? So would that be the idea?
Yeah, we start with monthly.
Do we have a lineup? Are you, if we find a paper we would like to discuss, should we send it to you, post it on BSI Slack channel? What's the recommended method?
I think we'll post it in the Slack channels. Okay. I did that before, try to get everyone's input to say which paper are they interested in. So that
we already have a list of papers there.
Oh, very good. Okay.
So you can see if and any of them is interesting to you.
Great. Okay.
Okay.
Okay, then start. Nora gave us a big start.
All right. Thank you, Jean. Thank you, Jess, and thank you, Armin, for organizing this presentation, and rather it's a discussion this morning. And we chose this paper published in Nature Genetics last year, a global overview of pleiotropic and genetic architecture in complex traits. And my background is molecular biology and biochemistry, and then turned neuroscientist, and also human genetics. So to start, this paper talks about pleiotropic, and we need to understand what pleiotropic means. So the idea is that pleiotropic means pleon is more and tropic is way. So it's how one single gene can be associated with multiple traits. And why is this important? Well, this is important because if one single gene is defective, then we can see that there may be diseases, several tissues can be affected. And in this case is the disease is Sickle cell anemia, which is beta globin and affects multi-organs, it's a blood condition that the hematocrites are very like sickle, so they die, they clot, they form blood clots, and so it can occur with heart problems, liver problems, brain problems, eye problems. So they have a multi-system problem, and so there will be multiple traits associated with one single gene. And I can tell you, in my family runs another mutation in the same gene of the beta globin called beta thalassemia, which is the Mediterranean blood, and we have a really mild form. My dad had it, my cousins had it. I don't have it, so in the same gene, different SNPs of different mutations can affect also different traits. So I think and then there are other diseases like PKU, that is a defect in phenylalanine hydroxylase, and we know affects the brain and multiple systems as well. So the premise and goal of the studies. So the idea is that in the past 15 years, there have been a large number, over 3,000 GWAS, genome-wide association studies, and with reporting more than 1,000 traits and tens of thousands of genetic risk variants of the single nucleotide polymorphism, or SNPs. And the idea is that diseases like schizophrenia, which we all study, are polygenic, mean multiple genes are associated, multiple SNPs are associated, but each SNP is of a small effect. So you have to look at the complexity of these disorders and looking at all the SNPs together. And Jim will talk about heritability later, but just to know that these obviously are highly heritable disorders. However, what they didn't know until now is whether all these genetics variants were associated with one, many, or even all traits that have been associated with human disorders or human conditions. So what they decided to do is they have this website, and the Atlas website that I is in the slide here. But basically, they have cataloged over 4,000 GWAS studies, and using the statistics of those, and those are several different studies, including two from the UK, several from the UK Biobank. And basically with these studies, they have been associated genes and SNPs have been associated with unique traits that I'll describe in a minute. And then those traits are classified in 27 domains, and I'll show you also in the next slides. But basically, so they have this online database. I'm not running live on this, but if you want to see it, it's very, in the next slide, I show you how I search for the schizophrenia. So if you can see it here, this is the Atlas of GWAS summary statistics. So the website was in the previous page, and you can browse all the GWAS studies, multiple GWAS comparisons. That's what they have been doing here. And then the phenome is the phenotype associated with the genetics. So, and I put all the genetic, the domains that they have been described. I added here psychiatric, and I said mental and behavioral diseases and schizophrenia. And here you can see how many studies you can check that have these associated keywords. And you also can select by year, but basically this is a very valuable resource if you want to do kind of looking at all the GWAS studies for one disorder. And then in the next slide, I wanted to tell you that, of course, the 4,000 GWAS are in the online in their atlas. But in this study, they decided to focus on those studies that had the highest number of subjects, over 50,000. And this is to avoid, errors. And also they have, so with those imposing the large size, the sample size, they have 558 GWAS. And it's also, those have 558 unique traits across 24 domains. And here is from the supplementary figure, and they show, I just have it on the screen, some of them, and you can see they are associated with, they give you the number and the associated traits, and this, and you can see even how many controls and cases are per study. So again, this is from the supplementary figure of the paper, but you can find also that information online. So I'll start with the first table. And what they did is they count and also determine the proportion of pleiotropic traits associated loci, genes and SNPs and gene sets. So I'll tell you a little bit more about how they did the study. So basically, they only use the autosomes. So they only use the chromosomes except the Y and the X. So they only use those to focus the study. And so the length of the genome, we know is 3,000 bases, but in here, 3,000 bases, 3 billion bases. But in here, because they don't count the X and the Y, then you have a little bit less. And so that's the whole set for them. And then they count the genes. Instead of 20,000 genes, they have 17,000. And they count us the SNPs being 1.7 million. And they count gene sets. And they use MAGMA to define the gene sets associated with specific pathway or a specific trait. And then they look at of all the genes and SNPs that are associated with traits in those 558 GWAS studies. They found that more than 50%, 60% are associated with traits. So it's a large proportion is associated with traits. And from this, what they do, they understand the pleiotropic effects means that they have one gene associated with both multi-domain and single domain specific. So if you add these two, so you have the pleiotropic here, you have to add the domain specific and the multi-domain, and that you find pleiotropic. So it means that they are associated with more than one trait and more than one domain. And well, we'll see more about it. And again, and then the loci means the percentage of the genome that these loci, these associated traits, the associated genes and SNPs covered in the genome, which is high proportion, is 93%. And then they look at the genes and SNPs and gene sets. And I'll explain this more in a minute. But I wanted to show you figure 1. And in figure 1, what they show is essentially the traits associated locus gene and pleiotropic areas in the genome. And the first is the proportion of the gene density, the proportion of the genes that are associated with trait. And as we said before, it's more than 60% for both the pleiotropic and the multi-domain, but not for the traits and the non-associated. So trait means single trait. So it's only one trait out of all the possible 558 traits. So those are non-associated means that those are not pleiotropic or non-associated. And if you can see, there's not much difference in the gene length for all of these, meaning that they cover the whole genome. Presumably they are scattered, so it's not they are clustered in any specific area of the genomes, but are more scattered. And finally, what they show is also they calculated what is called the probability of intolerant of being intolerant to loss of function mutations. And I gave you the website where you can look at this. But basically, I tell you how we know about this is by exome sequencing. You can tell which variants lead to disease because they are incompatible And these genes, sorry, the genes are defined here as protein coding genes. And so the idea is here, this is another interesting paper that I gave it for Jean too, because she's studying Huntington's. So and it here shows like at the level of the nucleus, you have transcription, you have processing, splicing, and there can be variants between genes, at the DNA domains that are regulatory in the exon that codes for the protein. And in here it shows all the different, where the variants may be localized. And we are going to talk about loss of function, means that you have less mRNA because it's not transcribed or it's degraded rapidly, or the protein is truncated and quickly degrade. And Huntington's is the opposite. They have gain of function because it's CHG expansion and polyglutamine. And basically here I gave you the sequence of the codons, and usually for being problems in the protein sequence, these are all the amino acids, you have to have mutations in the first or the second codon. And so, and I don't know if we have time, but I can tell you the difference between synonymous and non-synonymous mutations. And I wanted to look at, give you another site that you can look at annotated variants from the exon sequencing that are associated with traits. So these are the ANOVAR website. So the web ANOVAR is annotation of variants that could be also useful if you find a SNP and you want to know if that SNP is associated with loss of function mutations. Anyway, continue with figure 1. Then they look at, the tissue specificity of the genes involved in associated with traits. And here you know that if they are all 50, they use GTEx and they have 53 tissues. And basically all the 56 tissues have increased association, and the highest is usually when you have more domains, as a function of how many domains these genes are associated with. So genes expressed in multiple tissues, obviously, a single gene that is expressed in multiple tissues will be associated with more domains and will have more catastrophic results if it's mutated. And the same thing they do with the EQTLs with GTEx, and they found also that the SNPs that are associated with more tissues are also, they have associated with more domains, mean more diseases or other conditions. And the second part of this is what they did is they look at the SNPs associated, the proportion of the SNPs associated and the location of the SNPs. And basically here, I forgot to tell you that in mRNA, there are upstream sequences, and then in a gene, you have upstream sequences that are regulatory, 5' untranslated region, 5' UTR, that are more important for translation. Exonic is when you have the protein sequence, the 3' UTR, which is what I'm studying, and there's a new protein that the protein I study now has been associated with schizophrenia. I have to tell you, gene is KSRP, and I'll send you the paper later. But they have different areas of the mRNA or the here, and intergenic, and they also have non-coding. But you can see, the intronic associated will be the highest in here by looking at the colors. And then in here, in the right side, they expanded this area to be sure that you can read between these different variants, and exonic will be the most associated So SNPs that are in the protein sequence will be associated with, more associated with traits. And if you want to look more at how they did the GWAS summary statistics, there's a previous paper from the same authors that explain the flow of analysis, and I just want to leave it on there. I don't know all of this, but basically it's give you a good overview of how they did the analysis in this paper. And I think I'm done, so I'm gonna leave it to Jess to talk about figure 2 now.
Yeah, I don't have slides, so I'm just gonna share my screen here and pop up the images. I think thank you, Nora, for giving such a thorough background on the methods and what this is trying to do. And one of the reasons I think this is such a good paper to review is because it has so many pointers to algorithms and resources and software that we can use ourselves when we're trying to annotate genetics or trying to understand what we're seeing either in the literature or in our own data. Something I thought was actually very interesting as I was sort of looking through this table, it's worth spending some time, as Nora did, sort of understanding how this table is laid out and what it means. But I was particularly intrigued by this idea that 67% of genes are associated with something. 80% of them are pleiotropic, 67 cut across multiple domains, and 14 of them stick to a single domain. But if you come over to gene sets, a given gene set is only 10% likely to be associated with anything. 90% of any given gene sets is going to be not related. So I thought that was an interesting distinction between these gene sets that we think of as being biologically relevant, but the genes are actually more associated. I don't know, Nora, if that struck you at all or if that made perfectly good sense to you.
You're right. I forgot to mention that. Yes, it's kind of how you define the gene sets. And I'm not familiar with Magma. We use Magenta before. I think it's we'll have to see, maybe the algorithm, but yeah, the gene sets, you would think there would be more associated.
Do you think it would be similar? Because the gene sets are usually defined based on what we know about biological interactions between the genes and what they do, but apparently they're not relevant for identifying associations with traits, at least in this case, which is
well, maybe the genes are very large. The genes are very large, and so it's hard to associate the whole set with one trait.
Yeah. So
maybe when you go to the gene level and SNP level, yes, but when you go with large sets, they may not fit the trait perfectly.
Yeah, exactly. It fails to rise the level of significance in that case.
Sorry, just add one point. They mentioned later on one of the differences between gene and gene set. One is that they use the enrichment test for the gene set. So the gene set size does affect, what's the power you have to identify that. And another thinking I have is that gene set is basically a subjective definition, right?
Very much so. Yeah.
Yeah, it's like you can define this one as a set of genes and for particularly functions, define another set. So that may be also involved that our now incomplete knowledge regarding genetic functions to defining the gene set. That's what I thought. Maybe that is a reason for such a big discrepancy between gene set and genes.
Yeah, the number of gene sets are very small. That's if you look at the table. Sorry. I remember that looking at the table, the number of gene sets are very small compared to the genes. Okay, so that's maybe so there must be very large gene sets, and in order to fit a trait, you may need more, how you say, more granularity. You don't want to fit the whole thing. You want to fit,
Right.
individual genes. But that's a great point, both of you. Yes.
Yeah, I think it's worth considering. But I think, Jean, you're onto something there with sort of the issue of gene sets in many ways being a top-down distinction. There's nothing that they're very subjective in a sense. I mean, they're based on the biological knowledge, but they're based on our biological knowledge, which is not perfect yet at all.
Yeah, actually, I think they. Sorry to interrupt.
No, please.
Just adding like one point. Actually, I think they mentioned in the paper that they did this genetic correlation analysis. Then they did some clustering analysis based on this genetic correlation profile. And they found that the traits like aggregate or clustered in a different way compared to how they just like define the domain.
Exactly.
So, yeah. So basically, it's just like it's like how you define and how they mean like they will like organize themselves in a biological way, basically.
Yeah. Which is exactly the point I was going to make about figure 3. So no, this is because again, these domains are sort of a priori distinctions and clusters and categories that that we think of as being meaningful. And what they were trying to point out in many cases was that the genetics are not necessarily the same, and some of these domains are perhaps more biologically driven and others are not. So what they were talking about here was really looking at across all of the traits, all of their, there are 558 traits. Some of them fall into this domain of activities. Within activities, how significantly correlated are those different traits within a domain? And then this is really, how many of them actually seem to pass the significance test. And you'll notice that here in cognition, you actually have a lot of the traits seem to be genetically, even though there may be very different traits within this cognitive domain, they may be, they're much more genetically correlated than perhaps I think this is gastrointestinal traits, or the ear, nose, and throat traits, which seem to have a few that are very significantly correlated, just like activities, but there are many that are just barely not. So it was an interesting breakdown this way to sort of see which of these, because these are very different categories and domains. The difference between social interactions and skeletal traits. I wouldn't even put those on the same graph at all. But I think it's very interesting from the GWAS. That's what people are publishing. Some people are publishing on genetics of skeletal things, traits that could be grouped as skeletal function or respiratory function or reproductive function, as opposed to cognitive or psychiatric or the genome of social interactions. And then this is now looking at the cross-domain analysis or correlations, and of course down the diagonal, I was sort of like, wait, why is it not all ones? But of course, this is a cluster of traits. If you look at, for example, cognition, the cognition pairing sort of represents the fact that you had a fairly high cognitive correlation, but it's not going to be 1 because the different traits have very different genetic backgrounds. So within a domain, you have some variation pairwise, but then across domains, largely not a lot, except for environment and neoplasms. And a few others. So this was actually very interesting to show that even though within a domain we may sometimes not see very strong evidence that the traits within a domain are genetically related, we're certainly seeing that the traits across domains are not particularly related, except for a very few here and there that are well worth looking at. Did anybody else have comments or questions about that?
And also, if you look at psychiatric and immune, and this is something that Jean knows well about the MC, the major histocompatibility complex genes that affect, not only psychiatric illnesses, but immune systems.
Yeah, that's your little spot right here. It's just barely getting across. So yeah, psychiatric has some relationships. Reproduction has some relationships. And you can sort of see where these seem to be related to each other. With the star is the pairs of domains in which the majority of pairs are significant or are significantly negative.
This was negative. That's what's kind of weird. Why the pair correlation will be negative? Oh, you know why it's negative? Because when you have psychiatry illness, you don't have autoimmune diseases.
Oh, that's true. Yeah, you tend not to get rheumatoid arthritis and diabetes.
Sets the same genes, but they cause different traits.
Mm-hmm.
So they are not. And I think with neoplasma too, if you have maybe a schizophrenia, you don't have as much likelihood of having a cancer. So there are some interesting correlations, even though you in the immune system you have the same type of association, but with different consequences because there are different SNPs.
If I'm reading this correctly, you do have, I'm not sure what was in the activities domain, but you do have genetic, negative correlations for social interactions and cognition, which we also knew. Within psychiatry.
Someone says something in the chat. Sorry. Well, we can.
There's a question from Rogers. The 0.5 genetic correlation is not enough to claim genetic relation. So in general, my answer is 0.5 genetic correlation is high. That means that you consider these two traits are related.
Right, but what we're seeing here is not the correlation, but the proportion of trait pairs that actually have a significant correlation. Mm-hmm. So this 0.5 here, I think, is basically showing 50% of the pairs between, metabolic and skeletal had a significant correlation. And these are up to 0.75, which could be very, very high. But then we've got negative correlations in here as well. So this is a proportion that's, more than 50%, but negative, which is interesting to try to read. So they're trying to look at, this was sort of, are these domains actually all independent? And as expected, they're not really, though in some cases they're negatively correlated with each other. And then they come down to look at the distribution of minor allele frequencies across these different findings, across the different genome-wide association scales. If you look at the lead SNP, the SNP that was actually tagged as relevant, what is its minor allele frequency? And generally speaking, those low minor allele frequencies are overrepresented, and they highlight that this is in one way saying maybe the rare SNPs are actually having undue influence, but it could also be that the statistics get wonky when you're down at that low a number. Now, they did try to limit this to genome-wide studies where you had 50,000 people, which gives you at least a decent shot at getting a decent number of people with the very low minor allele frequency alleles, but it's still problematic. And this is really, this is sort of the projection of this, if you looked at it this way. And here you're seeing sort of the contributions of the different domains to this overall distribution, that our immunological folks really have some very low frequency SNPs. And then as you get down to endocrine and let's see, I think this one is endocrine and maybe this one is muscular, you're starting to see that they've organized it based on the overall proportion of lead SNPs that were a very low minor allele, and then how that falls off. And then again, the rest of this is largely the distribution of what these SNPs are actually doing. Where are they on the genome? And again, this is sort of showing the overall proportion that a number of them are intronic, but there's a number of them that are intronic or and non-coding RNA intronic. And then you have more of a distribution in the upstream and exonic. And they argued for some enrichment of the exonic aspect of the genome. Now, this PIP business, this was an interesting analysis. I wanted to say a little bit about this. This is where they use this fine mapping. That's the name of the name of the algorithm or the name of the procedure and the name of the software as well. And there are a number of algorithms that will do fine mapping. This is the one that they used. And what that's trying to do is really spread out the probability across the across the SNPs that are nearby, because we know that with genome-wide association studies, we're not looking at every possible, I mean, with genome-wide association studies, and certainly with the ones that were published here, we're not looking at every possible SNP. These are not whole genome studies. These are genome-wide. So you're picking tagging SNPs, and then you're doing your best to infer what that part of the genome is actually, that is being tagged, is actually doing, and which variants it actually is. And so there are, FindMap uses a Bayesian kind of approach to sort of say, okay, if this was the tagging SNP, what is the probability across the other variants that are nearby? And where do we actually, when you look at the posterior, the whatever PIPS was, the posterior something or other.
Probability of inclusion.
Yeah, posterior. Where did it go? Here we go. Posterior inclusion probability. So should that be included with the locus that was considered as part of the lead SNP? So you're looking at other credible SNPs around the lead SNP. So this is number of SNPs in the fine mapped region, and then how many were actually credible and what that actually does. And again, looking across here, the bulk of the genome is non-coding. The bulk of the lead SNPs are non-coding. The bulk of the, of the SNPs in the fine map regions are non-coding. But eventually you do actually start seeing some of the exonic and intronic. And they were basically arguing that a surprising number of the SNPs in both the fine mapped and credible and lead SNPs are actually really involved with the chromatin and the expression QTL. So the quantitative trait loci that affect the expression of the gene and the areas that are affected, either affected by active chromatin. So again, having to do with wrapping and unwrapping the genome so that it can actually be expressed and be functional, is what these seem to be these seem to be overrepresented. I think that was the point of all the math in there. And I think the rest of this comes down to you. Now we've sort of dealt with what are we seeing in terms of the genes? What are we seeing in terms of the genes related to the actual traits? What are we seeing in terms of minor allele frequencies? And how does that break down across these different trait domains? And I think, Jean, it's up to you to bring us home, right?
Okay, so I probably just add a little bit of these sections that I can show my screen here.
Yeah, go ahead.
Oh, okay. Share. So when I'm reading this figures, that's really a lot of information there. And then first I thought about, okay, look like the probability as well as effect size from the SNPs, which has really low minor allele frequency, is high, right? And then how that affects our general analysis, because usually we're thinking if we do have looking at genetic mutations effect, we kind of have a QC step put in, the minor allele frequency smaller than 1%. We kind of ignore them because that is just require large of numbers, samples. But here that looking at, okay, the effect size is much higher in the minor allele frequency less than 1% SNPs. So does that going to change a little bit our research strategy or how that affect us? That I'm just beginning to trigger me to thinking, okay, so 1%, like a minor allele frequency less than 1%, that is where all this is up of the square center, the effect size going up. The rest of above that seems like a flat. So effect size higher than 1% to 50% is very similar, but the effect size less than 1% is dramatic differences. For how we take up that information in this figure. And then the other thing is talking about the leading as well as credible SNPs, which are usually causal. You can think of credible SNPs like a more likely to be a causal SNP, causal effect, right? So this causal effect, and then the more of, I think the proportionally more in the location, I think the green is
I think
intronic.
they are like more axonic.
As my reading is increased in the intronic regions, right?
I think it's like the other way. As far as I can remember, it's like the trait-associated SNPs are like enriched in the intronic region. But if you compare the lead SNPs with the trait-associated SNPs, so basically you are comparing lead SNPs against the trait-associated SNPs. They are more enriched in the axonic regions.
Yeah, I think that's what they were their point they were trying to make there, though it's a little bit tricky to see.
Yeah, so overall, the associated SNPs is in these intronic regions, more enhanced in these regions. But if you compare leading with trait, that is this comparison of these two, where the switch locations, right? That will be the exon regions is more above. Okay, so that is like if we look in the more causal or more large effects, we're still in the exon regions.
Yeah,
but
I think
overall,
that's
right? Overall is in this gene in the genic regions, right? Because I think just you mentioned that large proportion is in the non-coding regions. However, if you're looking at non-coding regions, that the intergenic is reduced, but intronic region is increased. So that part. So even though it's non-coding, but it's within the gene, it's the intronic regions are more likely increased for the causal as well as the leading SNPs.
Yeah, and intronics, okay, and a SNP in an intronic region affects the splicing, how you put together the protein. So if it is associated with a trait, means that the way you splice the exons together has been altered. That's it doesn't change the proteins. It does change the protein sequence, but it's because you include or exclude exons.
Right. Yeah. So while I read here, the thing struck me is that before with the previous publications, kind of like the intergenic region is getting more and more attention, but this paper is really leading to is not all non-coding regions, but the more of intronic non-coding regions. So that is what I take from here is, okay, we know they're not very important non-coding regions, but proportionally is intronic non-coding regions. Is that fair to say?
I think the issue is that they map all to the genes, and genes are defined here as protein coding genes. So the focus was in those 20,000 validated protein coding genes in Ensemble. So that's the reference. So the non-coding regions here will be in a gene, in a protein coding gene will be the upstream region, downstream region, and the intronic region. So it's the 5' UTR, 3' UTR, and the intronic. And because in a SNP in intronic region, it's associated with a trait, means is because it affects how the splicing is occurring.
But when they refer here, this term intergenic region, I thought this is outside the 20K genes. Right?
Yes, intergenic means between genes. Yes.
Right.
Yes.
So I'm just thinking a lot of SNP, the associated SNPs are in non-coding regions. But when we're looking at the credible SNP as well leading SNPs, where intertronic regions is proportionally higher. So right. So okay. Yeah, so that's saying, so give me some of this, like what is really important regions when we look, if we have to looking at the importance of SNPs, right?
I think their summary is that the trait-associated SNPs are enriched in intronic regions, but depleted in the intergenic regions when you compare them against the whole genome.
Right. Yeah.
So that's the observation. So basically, you have enrichment in the intronic region and have depletion in the intergenic regions.
Yeah. So that is making me think about this region is probably more get my attention, right? So that is when we looking at the treat this intronic region, even though it's non-coding, but really has a stands out. And then further is when we're looking at real leading as well as credible or causal SNPs, there where is extronic, this region stands out. So that's some information getting when we're looking at this GBUS results, what is the proportional importance of the regions. And then, so then we switch next topic is looking at SNP heritabilities. So this is the way to looking at, so using-
Sorry, Jean. Sorry, there's a question in the chat. I just want to point out.
Okay. Okay, I can look at, how do I read that?
So it said, the question is, are these results from EA only?
From
European ancestry?
Yeah.
Asking, okay.
European and Asian, yes. There are, let's see, the majority of the are European and Asian. But they map others. Let me see. They have some African. How do they do it? You know, the majority of the GWAS, at least in schizophrenia, is European and Asian.
So my question is, if they mixed the European ancestry and African ancestry, then how, when you present the Mali allele frequency result, do you think that could actually influence the result, and will that influence the conclusion?
Well, they impute against the known minor allele frequencies in each group. So they have that into account when they do the imputation. So I think it shouldn't affect that because they imputed. But yes, you're right. The minor allele frequency in Africans and Europeans are very different. But I think the majority were Europeans. Let me check the paper.
I think the question is valid because if we put everybody together to say, okay, this SNP in amandali frequency less than 1 or less than 1% or less than 5%, that is overall effect. But the 500 GWAS, I'm thinking majority are Europeans.
Yes, the majority are Caucasians. I think they refer to 1,000 genomes.
The panel, reference panel.
Yeah, the reference panel is 1,000 genomes.
Their imputation is no problem. They impute based on their own race groups.
Yes.
But when we try to get this summary results, right? Overall effect of this trait-related SNPs and then overall distribution of minor frequency, that probably more reflect the Europeans instead of other groups.
Yeah.
Yeah, that is good. That's right point.
Yeah, the UK Biobank might be overrepresented in this this work since they did a bunch of their own analyses on the UK Biobank as well.
Yes. Yeah, that probably reflect the more EA population too.
Very much so.
Yeah. So the rest of the paper is looking at SNP heritability, and for this is looking at, we always looking. The questions come up is, how heritable this trait will be, and in terms of how much samples you need to really identify a stable SNPs causing it as well associated SNPs, right? So that is looking at based on the SNP GBUS results to compute the SNP heritabilities. There are several algorithms you can do it. So this paper used 2 of them to do the comparison. One is using the LDSC, the other one is the sum heritabilities. So overall, the consistency is 0.77, So you can see the overall is consistent, but some group are some heritability give you a little bit higher than the average. But overall, I think, okay, a little bit consistent heritability computations based on 2 methods. And then, so I think the paper is presenting using the LDSC heritability estimations. Around 0.2 and 0.3, that's mostly likely we say. And I think the previous papers also have a comparison regarding heritability estimated by SNP, like this one, and heritability estimated by the family structures. Usually the SNP is estimated heritability is a little bit low, like here's 0.3, 0.2. But if we're looking at heritability estimated by the family structures, usually give you high, like 0.5, 0.7. So there is a discussion regarding how we align these heritability estimations using the SNP and heritability estimations using the family structures. So overall, the heritability estimates of SNP.
Sorry, Jing, I have one question. Is that right that for this this LDSC computed heritability, it's only accounting for the common variants?
Right, comments. Well, you can see, but if your SNP go down to less than 1%, technically it's not common anymore. When you have larger sample size, you can go lower than 1%.
Right. But I guess I'm saying that we still have lots of other genetic variants, which not taken into account.
Right, like copy number variation. Yeah. For that variance, this SNP heritability does not take account. This is only using SNPs, right? So it doesn't account other type of genetic mutations. That's true. So that is basically try to put the point that there are differences between SNP estimated heritability and the family estimated heritabilities. But this is only saying heritability estimated by the SNP. You can say, okay, there's a large range in these 500 traits from really low to really high. The highest one is like height. This gives you the high SNP heritabilities. But overall, there's consistency between the two methods. And then the key question here, the answer we try to answer is that, what is the relation between the heritability or the polygenicity, that is a computation, usually to say a number of SNPs being called for particular traits, and then the discovered abilities. So the higher you have more SNPs being identified and associated with particular trait, you have high polygenicity, and individually the discoverability get lower. So the higher here, then your discoverability getting lower. It's like, I think it's more of effect size, individual SNPs effect size getting lower. So in order to discover it, you really need a large sample size to have a discoverability. So that is basically saying that in order to say for some traits, maybe 50,000 sample is enough, but for other traits, you really need 100,000 of samples in order to have a stable number of associated SNP being discovered. So that is the information this graph is gave us. So for the complex diseases with much more large of a polygenic SNPs being identified, that we really need large samples. And then if you increase your sample size, you can say more and more SNP being identified, but it should be saturated when with sample size reach the discoverability allowed. So that is the information to get here. But also there are some trade-off. The current method I mentioned, well, this method here is still limited by the method we have. So that is basically the take-home message, the last message in this paper, just saying, how when we do the GWAS, how we consider the sample size is big enough. So the summary of this discussion is a paper. What I'm considered more of this large proportion of the treat is really polygenic. It's like a pleiotrophic, right? It's single SNPs associated so many treats and even cross domain of a treat. And then so when we have this GWAS results, and then because from my perspective, what I want to do is I want to leverage the GWAS results to pre-select the SNPs most plausible for particularly treat. What's the information we should get in, right? Is that this SNP because affect multiple domains? Does it make a higher weight in order to study it? Right, so like looking at, okay, this SNP has a cross-domain effect. This SNP location is intronic regions, exon regions. Is that more plausible to have effect and to put into as a target for us to study? I think the one missing part is...
I'm sorry, Jean, can you say that again? What does it make sense for us to study?
That's more of it has effect across domains, multiple effects. And also the location, right? Does the intronic regions and the exonic regions give a high priority?
Mm-hmm.
So that is basically initially I thought that maybe intergenic the region is, but right now I'm kind of switch my idea to more of intronic regions.
Yeah, no kidding.
Here, right? And also another is talking about the regulation effect. I think one of the plot showing the SNPs highlight in regulation effect. Where is that?
Can I add one more thing? Hello?
Yep.
I wanted to say that the intergenic region, we don't know as much because it's more regulatory and it's not as, functional. A trait, its gene codes for the protein and the protein participates in, organ function, tissue function, cell function. So for the others, the intergenic, or the non-coding, unless we know specifically what they do, it's hard to associate it with traits. And I think that's what it's telling you is that SNPs associated with genes, which are the minority in the genome, are the ones that are mostly associated with the traits.
But when we do the GWAS, that's not considered the location. So proportionally, if the intronic region is sent more, it's not really regarding or because we do not know much of the intergenic regions and we know a little bit more about the intronic regions. Right?
Yeah, but the intronic regions, again, the genes are 2% of the total genome. What I'm trying to say is the protein, the traits are associated with proteins, and the genes, they use protein coding genes. So the intron is in a protein coding gene.
Right. When we do this, this proportion, the percentage of this, right?
Yeah.
When you do this percentage, then that is not considered the gene coding genes. It's a genome compared to genomic percentage. So if that is the case, then proportionally intronic region is sent more when we're looking at the trait associated loci.
Yeah. And this is because the protein coding genes are the ones that are more associated with traits. And the introns are in the protein coding genes. Right. So, so I think it doesn't mean that the other SNPs don't have a function in terms of genetics. The only thing is that when we are talking about association with traits, in this case, we are biased to the protein coding genes because those proteins are associated with traits.
Right. Okay, I get your point. This is more in terms of effect size the protein coding genes likely have a large effect size on the trait, particularly selected.
Yes.
And then the yeah, okay.
Yeah, I think one way to interpret is that, because the traits, you know, the traits, even any trait, you need to associate it with a protein. And that's why.
Does the paper at the end talking about the most pathways which are linked to highest cross-domain, multi-domain connection, the gene set is the one which regulatory effects? I think there's one that we're talking about, the regulatory effect.
Yeah, I think the most enriched gene set is the regulation of transcription.
Right. Yeah. That is common. So I think that is given information. Regulation of the transcription is more of a common to affecting most of more than half of the traits.
Yeah, you're right. Transcription factor, without transcription factor, so transcription, you don't make a protein, so it's the upstream.
Right. Okay, so that is one point. If looking at not exactly like trait specific or domain specific, it's a more cross-board effect is the one, the regulatory pathway or gene sets.
Yes, and because transcription factors can activate multiple genes, at that level you have one single protein or one single protein can activate multiple genes. And so we don't have transcription factors are very few in the cell, so they have multiple roles for different genes. So at least, like CREB, the CREB will activate many, like BDNF, it activates a lot, microRNAs activates a lot of, so one transcription factor activates a lot in an activity-dependent manner, can activate a lot of pathways.
Yeah, okay.
That's how I interpret in terms of the biology of why they have those.
Yeah, that definitely makes sense, right? Okay. I think that's about 1 hour.
Oh, yeah, goodness.
Yes.
We're over.
That's a very good discussion. And we basically, I think, is there any burning questions you want to ask regarding this paper?
Can I ask 2 general questions?
Yep.
The first one might sound silly, because in the first table you showed there is like around 35% of gene that is not associated with any trait. So I just wonder, is it possible that there's genes that is not associated with any trait? And my second question is, after we've been doing so many GWAS and as this paper shown, loving the result from the GWAS, what do you think is the next step?
Okay, so the first question wasn't about table 1?
Yeah. So I can show where is the table 1. It's this one, right? So they're talking about this non-associated, this one, genes. So
yeah,
34% genes are not associated. Yeah. So that is, what do you think, Nora? Are they really not associated anything?
Well, okay, they don't define every single trait.
That's right. We've got a limit of human effort here too.
Yeah. So it's not that and again we define association by the mutation. So if it's associated with trait, we only know it's associated when the absence of or that SNP or that gene causes a change in the trait.
without being fatal.
Compensatory traits. There are multiple genes that are associated with the same trait. So it's hard to say that they are indeed not associated, or that other genes or other SNPs are compensated for that. So the absence of that gene may not be as important, because there are gene duplications. There are, when we how many protein kinases? 4. We have 5. Okay, so CAM kinase is 1, 2, 3, 4, at least 5. So the absence of one other may compensate for that. So you don't see the impact in the trait. It doesn't mean that normally it's not associated. But in order to know it's associated, the only way that you know it's associated is when something goes wrong, and then you see an impact on the study. So all these studies are mostly disease associations, okay? So diabetes, muscle skeleton, cancer, immune system, all of these studies. So in order to really see it, it depends, is how the GWAS was done, and whether, or what specific traits, how traits were defined. So the fact that this says they are not associated, I will be cautious to say that totally they are not associated. There may be other compensatory effects. We see that in mouse knockouts. So if you have a knockout of one protein, another protein in the pathway can compensate. You only see really when there's no compensation, then it's when you see a specific effects of the gene. And traits are determined by more than one genes, several, hundreds of genes may be important for each trait. So I wouldn't think the non-associated with a single trait, I would say, is probably the way it is operationally defined, each trait. And again, it's because the way they look at diseases, what is the study is using to define the trait. What was the other question? Sorry.
What will be the next step after GWAS, right?
Yeah.
Oh, okay. Well, I don't know. I can see this. Okay. The problem with the GWAS studies before is that you had, if this gene is associated with this, the other gene is associated with another illness. And I think what the beauty of these studies is that they are looking at multiple GWAS, a lot of GWAS. And that's when you can say across GWAS, when they have similar traits that have been studied, you can see how many of these genes are associated with multiple traits. And before, individual GWAS study were, okay, so we all look at schizophrenia or bipolar disorder, and we look at all the genes associated with schizophrenia, with all the traits of schizophrenia. But as we discussed the other day, the genes associated with schizophrenia are also associated with bipolar. 60%, 68% are also associated with bipolar. Are also associated with a lower percentage with major depression. So when you said genes associated with schizophrenia are not only associated with schizophrenia, are associated with a host of other traits like cognitive function. There are many more. So you cannot say, the beauty of the study is not disease specific, but trait specific. And that's moving forward, to an understanding of genetics in human populations, is that you have to say, the gene as pleiotropy, one gene, multiple diseases, one gene, multiple traits. And I think that's what this study is using. So I think we'll have to redefine how we look at disease-specific SNPs in the context that the comorbidity, and that's another issue. if you have a same SNP associated with or gene associated with multiple traits, you will know, for example, schizophrenia and substance use will be comorbid. They are depression and substance use is also comorbid. So many of the traits like anxiety, okay, will have depression, substance use. So it shows more the complexity in a global. So if you look at from the top of the tree and you look at the branches, instead of looking at one single branch, and I think that's the beauty of this study.
But one thing, especially in psychiatry field, is the definition of the disease, the phenotype of the disease. Do you think that could actually restrain the power of the GWAS?
Can you repeat? Sorry.
Yes, is the definition of the psychiatric disorders. I mean, whether the symptoms or the symptom basis. But do you think this phenotype, the definition of this disease phenotype, could actually restrain the power of GWAS?
Restrain the power of GWAS?
Yeah, because a lot of SNPs you see they are responsible for, let's say, schizophrenia or bipolar. Is it because they have the same symptoms?
Yes, and that's the arc, you know, the art docs. The idea is that you, instead of looking at diseases, you look at domains. And this is, you know, it's the idea that diseases are in continuum. You know, you have schizophrenia, schizoaffective, bipolar, and more. So it's a spectrum. It looks more as a spectrum of the illness. It's not it's too much classification, you know, it's looking at diseases in terms of domains and traits rather than, yeah.
Yes, but most of the GWAS they include in this study, they are binary, right?
Yeah.
No, they have non-binary and binary.
Yeah. Oh, okay.
They
Marvelous.
have both. If you look at the, okay, where are the, in the...
Yeah, I think they have both continuous and binary.
Yes.
Yes, but they didn't show the result of the comparison.
Oh, no, yeah. Comparison is not included in this.
Unfortunately, I have to hop off.
Oh, okay.
It's a,
Yeah, we're over time, I'm afraid.
yeah. But that's a good discussion. That's a very good discussion for the field.
We can
Yeah.
certainly take it to the Slack channel as well to see if we can have a conversation there more asynchronously.
Right. Can post any thoughts on the Slack so then we can find time to respond to each other.
Mm-hmm.
Okay, sounds good. Thank you, everybody. Yeah,
Thank
great
you.
discussion. These are very complex topics, so to be continued.
Absolutely.
Right. Okay. Thank you, Nora. Thank you, Jen.
Bye.
Thank you.
Bye.
Thank you, everyone.
Thank you, Jean.
Thank you.
Thank
Thank
you,
you.
Jiayu. Thank you, Armin. And thank you, everybody, for listening. Bye-bye.
Thanks. Yeah.